Thank you for the opportunity to present an update of recent activities for MetaZooGene SCOR Working Group 157. I'm Ann Bucklin, the chair from the University of Connecticut, and I'd like to acknowledge my vice chairs, Katja Peinenberg from Naturalis Biodiversity Center and Senia Kosovakova from the Shurtzoff Institute of Oceanology, and Todd O'Brien from NOAA Fisheries is available for Q&A in person, virtually, I believe. MetaZooGene includes 23 members from 19 countries. We are very fortunate to have a genuinely global extent of our engagement. Our primary focus are 8,000 species of holozooplankton, 8,000 and counting, 15 phyla, 32 classes, remarkable diversity of animals that drift with ocean currents. Our primary goal, integrated molecular morphological taxonomic analysis of marine zooplankton biodiversity throughout the global ocean. Our terms of reference are listed here to create an open access web portal for DNA barcodes, design an optimal DNA barcoding pipeline, and examine best practices for DNA metabarcoding of marine zooplankton biodiversity. We've been very fortunate to have in-person working group meetings despite the challenges of recent years. In 2019, we met in Gothenburg, Sweden. In 2020, in San Diego. And in 2022, in Dublin, Ireland. You can see here a picture from our symposium with early career scientists. The Working Group 157 is built around molecular environmental genomics approaches that are transforming our ability to analyze and understand marine biodiversity. The approach is called DNA metabarcoding, which entails analysis of environmental samples by high-throughput DNA sequencing of short sequences called barcodes, yielding millions of DNA sequences in parallel and allowing detailed large-scale analysis of complex samples. Now you can see here the steps in this complicated process, but the power of a metabarcoding is the rapid detection and identifications across the taxonomic spectrum. So you can see here on the right side, a picture showing group identification based upon the barcode of life metabarcodes, but there are other barcodes that I'll talk about that allow species identifications. The primary and central contribution of SCOR Working Group 157 is the MetaZooGene Atlas and Database. Working Group member Todd O'Brien from NOAA Fisheries has pioneered this effort, designed a web portal, atlas, and database for DNA reference sequences for marine organisms. The data are mined from NCBI GenBank and the Barcode of Life database, and currently unpublished data from MetaZooGene members. The initial focus for the MZGDB, as we call it, was the barcode region of mitochondrial cytochrome oxidase 1, CO1, but MetaZooGene rapidly expanded and now includes gene regions for a broad range of marine organisms, fish, mammals, turtles, seabirds, phytoplankton, and protists. Includes more than 1.2 million sequences for multiple mitochondrial and nuclear gene regions that are used for DNA barcoding and metabarcoding. The MetaZooGene Atlas and Database was introduced and advertised in a 2021 review paper published in Marine Biology. Now, this focused on the CO1 barcode records, which were first. But the paper has achieved its purpose in calling attention to the resource, the MZGDB. It's been highly cited in the peer-reviewed literature. Moving on, Todd and other Working Group 157 members, including myself, followed up with a book chapter. This is in the Springer series, Methods in Molecular Biology by Invitation, and it's been accepted for publication in 2023. Now, the goal here is to provide detailed instructions to users who are seeking to use the protocols and procedures that are associated with the Atlas and database. These tools are unique for analysis interpretation of DNA metabarcoding data. So the chapter explains how you undergo the unique search and access functions, barcode records, access to species lists and collection metadata. But it also explains, importantly, the search and mapping functions, which allow selection of data by taxonomic group and geographic region. And this is essential because it allows assessment of completion of the data records. Now, this completeness assessment has been invaluable for guiding and encouraging ongoing efforts towards completion of the reference database, which of course is a primary goal of MetaZooGene for multiple genes for many groups across marine organism diversity. MetaZooGene has a primary goal of peer-reviewed publications. These are directly related to the Working Group 157 terms of reference. And of course, it's important, I think, important that the contributions from the working group members are of direct benefit to them as mid-career, early career scientists. That means peer-reviewed publications. The current totals for MetaZooGene are 42 papers with member co-authors. Of these, 13 formally acknowledged SCOR, 36 include early career co-authors. For 2023 to date, 6 papers have been published, including 1 acknowledging SCOR, and there are several additional publications, including the chapter that I just mentioned, that are accepted or in press and not listed here. MetaZooGene was approved as a UN Ocean Decade Action a couple of years ago in association with the Marine Life 2030 program. Now, the UN Ocean Decade is a huge multi-dimensional activity, which is providing valuable opportunities to the working group members and colleagues who offered to serve as project representatives. Now, they're engaged in global scale communication, coordination, collaboration for research, management, and outreach. It's turned out to be a wonderful opportunity for those interested in exploring these avenues for further impact and communication. MetaZooGene has important plans in place for 2024, which we expect will be our 5th and final year of formal SCOR sponsorship. 2 activities that are central to our terms of reference are explained here. The first is the MetaZooGene Intercalibration Experiment, MZG-ICE, which is related to best practices for DNA metabarcoding. Now, MZG-ICE is involving 10 teams from Working Group 157. They each provided DNA extracted from one zooplankton sample of their choosing. You can see the global extent of this effort. DNA aliquots were exchanged among all the groups, and each group sequenced 4 gene regions for all 10 samples, and were carrying out centralized bioinformatics and statistical analysis of the assembled data. This is going to allow evaluation of the reliability and accuracy of DNA metabarcoding, regardless of the details of the protocols, procedures, lab conditions, and whatever other individual activities that are inevitable with research laboratories. The research will be the results will be summarized in a peer-reviewed publication, and they'll also be presented at an upcoming meeting. Now, that upcoming meeting is the 7th International Zooplankton Production Symposium, and it's going to be held in Hobart, Australia during March 2024. Several sessions and workshops will be chaired by Working Group 157 members. Many presentation abstracts have been submitted by members, colleagues, and many early career scientists, and we'll also be holding our annual 2023 working group meeting during the production symposium. So we're really looking forward to this opportunity to get together again, evaluate progress, and look ahead to the future. With that, I would like to thank you all for your attention. I'd like to acknowledge the invaluable contributions from all members of Working Group 157, and also acknowledge the amazing contributions of our early career scientist colleagues. We also appreciate the support from SCOR and hope we have taken good advantage. Thank you for your attention.